The question appeared in 2020 NEET. The question from cell cycle and cell division, it appears to be with ambiguity, but it is not with ambiguity. It is tricky with intricacy as far as the cell division topic and the question concerned. In this question, some dividing cells exit the cell cycle, enter vegetative inactive stage, full stop. This is called quiescent stage G naught, full stop. This process occur at the end of. So, in this definition given in question with regard to the G naught. And in cell cycle, the interphase and it is the division phase. In interphase G1 S G2 and in division phase it is the M. The G1 it is the decision stage, whether it is proceed to a stage or to exit G1 enter G0. So, that is a decision stage, and S it is synthetic stage. The synthetic stage it is DNA replication occurs, doubling of DNA occurs in case of S, and in case of the G2 commitment for cell division, that means it is ready for cell division and enter into the next stage it is M. The M stage is said to be completed only after cytokinesis, and in case of the G1, some adult animal cells do not appears to be exhibit any division like it is the hot cells, and some cells occasionally divide to replace the cells which have been lost during injury or cell death. So, cells do not divide further, that is not at all divide like it is hot cells, and occasionally divide to replace the cells that lost due to injury, exit G1 enter G0. So, at the end of the G1 it exit G1 enter G0. When the cell to exit or to enter, first it has to enter into what it is a stage, what do you call it is the decision stage, that is the G1. When it receives the go ahead signal, it enter into S. When do not receive the go ahead signal, it enter into G0. G0 it is metabolically active. There is no proliferation of this cell unless on to do situation. Whenever there is a requirement, it exit G0, enter G1. So, G0 is not at all said to be a termination stage, it is a suspended stage. So, whenever there is a requirement of the cells, it exit G0, enter G1. When there is no requirement of the division, it exit G1, enter G0. So, G1 it is a decision stage, every cell has to enter into the G1. So, in the diagrammatic representation of the text, it exit G1, enter G0. So, in this the right option it is the fourth option. This question appeared in 2020 NEET from Botany, the topic it is biotechnology. Seems to be with ambiguity, but the question it is a tricky with intricacy. As far as the question concerned, identify the wrong statement with regard to restriction enzymes. Nowhere he mentioned the function of restriction enzyme. With regard to the options, they are useful in genetic engineering. Second, sticky ends can be joined by using DNA ligases. Third, each restriction enzyme functions by inspecting the length of DNA sequence. And fourth, they cut the strand of DNA at a palindromic site. A restriction enzyme belongs to the enzyme group that is nucleases, a large class of enzyme that belongs to hydrolases. As you come across this in cell structure and function, lysosome. Lysosomes having nucleases that belongs to hydrolases. In case of the biomolecule, hydrolases that belongs to third class. So, restriction enzyme belongs to it is third class according to IUB system of the classification. and the restriction enzyme inspect the length of the DNA sequence, it is right. It first recognize the palindromic sequence, it cut the strand at palindromic sequence in the middle of the palindrome or just away from the middle of the palindrome. If it is just away from the middle of the palindrome, what do you call the cuts? It is staggered cuts that results in sticky ended fragments. Exactly in the middle of the palindrome results in blunt ended fragment. So, restriction enzymes cleavage of the DNA molecule results in sticky ends or blunt end, but most of the cuts which are sticky ends, that is right. And they are useful in genetic engineering. Yes, they are useful in genetic engineering to cut a particular sequence. Second, the sticky ends can be joined by using DNA ligases. As DNA ligases, which any in the two DNA molecules are two fragments and that belongs to IUB system of the classification, that is 6th class, as the restriction enzyme belongs to 3rd class, as ligases belongs to 6th class. So, in a given option, the sticky ends can be joined by DNA ligases not at all related to restriction enzyme as per the options, as well as it belongs to hydrolases, that is 3rd class, ligases belongs to, it is the 6th class. So, right option it is second option only.